Before moving into the third method of viral cultivation for animal viruses, we'll see certain part of the second method, that means tissue culture, in which how do we detect the viral growth in a cell culture. Basically, there are two methods usually adopted. One is the plaque formation, as already detailed in the bacteriophage cultivation. There is a clear zone formation in the monolayer of tissue or cell culture, and this zone is what we call the plaque, and these plaques are observed as an indication of the viral growth, and this plaque counting is used to estimate the bacterial number, and the plaque morphology and other features of the plaque is studied for the characterization of the virus. That's about plaque formation, and that concept is what we call the plaque assay. And the second one is what we call the cytopathic effect, and in certain cases the plaque formation may not be that visible. In that situation, we cannot directly physically detect the presence of viral growth. In that situation, we have to go for a microscopy. So under microscopy, you can observe for some degenerative changes of the cell that are linked with a multiplication of certain virus, and these changes, these damages formed, that damages created in a cell as a result of the viral multiplication is called cytopathic effect, right? And each virus will produce a characteristic cytopathic effect. So this characteristic cytopathic effects are used for the identification of certain viruses also. So here you can see the plaques formed, the clear zones in a plate after the growth, and these clear zones indicate a viral particle and their multiplication. So these are nothing but the localized area of tissue culture plates where tissue destruction or lysis has occurred and seen as a clear area like a zone. And this could be clearly detected using staining, like staining with a neutral red or a trypan blue, so that you can the cells will be stained, and the area where no cells is growing will be observed as a clear zone, right? So that's about the plaque formation and detection using plaque formation. This is about cytopathic effect, which is a microscopic or macroscopic degenerative changes or abnormalities in host cells and tissues as a result of viral growth. You can see the normal tissue and those which after a viral growth. So these are the two very common methods for the detection of viral growth in a tissue culture, right? So now we are moving into the third method of animal viral cultivation, that is inoculation of the virus into an embryonated egg. So embryonated egg, it's a cheaper method, comparatively cheaper and easy method, and widely used method for cultivation of virus. It's suitable for the even isolation, identification, and other purposes like vaccine production and all. And an embryonated egg will have different locations, and different viruses are inoculated at different sites in the embryonated egg. You can see sites like amniotic cavity is there, yolk sac is there, chorioallantoic membranes are there. So different sites are there, and onto different sites you can inoculate different types of viruses, and accordingly you can characterize them also, right? So the different routes or different sites are denoted in detail as chorioallantoic membrane, amniotic cavity, allantoic cavity, and yolk sac. Into these different sites you can inoculate different viruses. So if we are using CAM, usually they produce a visible lesion called POCK, and these pokes could be used for the detection. Otherwise they form certain inclusion bodies or edema or some kind of abnormality. This could be detected on the chorioallantoic membrane as viral growth. Certain examples of viruses like herpes virus and pox viruses are cultivated in on CAM. Amniotic cavity, usually again used for cultivation of certain viruses like mumps virus or influenza viruses, right? uh, the virus to be cultivated is inoculated into this cavity where you will have certain fluids, and into that fluid the virus is inoculated and they'll grow there. And not widely used, but used for cultivation. And it's given a last point: newly isolated influenza viruses may require several passages before they adapt to growth by other routes such as allantoic cavity. That means these amniotic cavity is unique, and those viruses which are multiplying in amniotic cavity, if you take out and inoculate into another location or another mode of propagation, they'll take some time to become familiar with that. Again, certain viruses like influenza virus and Newcastle disease virus are inoculated into allantoic cavity. It's comparatively easy to inoculate and cultivate viruses. Then yolk sac, again one of the simple method in which mostly mammalian viruses are isolated using this method. Okay, so these are the different routes of inoculation for animal virus into an embryonated egg. And there are various advantages. Basically, this is less labor needed, cost effective, and maintenance is much easier compared to tissue culture or animals. Then free from contaminating bacteria and many other viruses, and used for virus for vaccine protection also. These are various major advantages. Then how do we detect the growth in inoculated egg? Those virus multiplying in the embryos or may not cause effects. Sometimes they produce some visible effects like pokes or some lesions. In some other cases they may not be produce such lesions. So the major signs are, or major symptoms of the viral growth in an inoculated egg is: one is there'll be the death of the embryo, or it could be a defect in the embryonic development, and otherwise it will be a damage that is visible as a lesion or a poke. So these are the major symptoms by which we can detect a viral growth. Then pokes are already stated are the lesions formed on the egg, especially on CAM, chorioallantoic membrane, after the growth of usually lysogenic virus. These lysogenic viruses will not lyse the cells, but instead they will be there, and in that case you can see certain propagated viral mass will be there, and they'll be visible as a lesion. And these viruses, they form these lesions, could be experimentally or that could be observed on the chorioallantoic membrane. And these pokes also could be used for estimation of viral growth. That means you can count these pokes and used for estimation. And here again you can use the poke forming unit as a measurement unit. Okay, so by that the different methods we have discussed. Now we will see certain methods used for viral quantitation. And in this part of presentation, I am not going to explain all those methods. Already we have discussed certain of these methods, and we'll see a few more methods. So these are listed like direct microscopy count is there, plaque assay, poke assay, which already we have discussed. Then viral neutralization assays, the endpoint dilution assay, hemagglutination assay, radial immunodiffusion, flow cytometry, ELISA methods, Western blot, and nucleic acid hybridization studies. So all these are used for the viral quantifications. So in which few of the methods we will see. This is a microscopic visualization of a tobacco mosaic virus. So by direct microscopy you can observe. Usually we use electron microscopy, electronic microscopy for electron microscopes for the observation of viruses, since the light microscopy cannot differentiate them. Then plaque assay already we have discussed. I am not going to discuss again. A serially diluted sample will be used, and these samples will be plated, and you can count the number just like we do in bacterial system. then you can see the plaque formation. And then the pork assay is also already we discussed just now. That is the pork formed on the chorioallantoic membrane will be counted and recorded. Then hemagglutination. This apply a process of hemagglutination in which the RBCs will be lies bind to the hemagglutinin glycoprotein found on the surface of certain viruses like influenza viruses. So they create a network, and this lattice formation leads to the agglutination. So if virus is provided, they will give you an agglutination. Right. So in a serially diluted sample, if you provide virus and the RBCs, based on the concentration of the virus, you will get the different concentration of agglutination also. And just like in Vidal test, how we detect a titer, here also you can detect a titer. A titer means a concentration of the virus up to which you can see a visible agglutinate. Right. This will give you an idea about that. If you have virus, the image B, the virus and RBC is provided, you can see an agglutination. You can also go for indirect method, that means hemagglutination inhibition assay, in which the virus is given and also the viral specific antibody is given, and observe for no agglutination. So in that case also you can go for the detection. Then endpoint dilution is done in case of tissue culture method, in which half of the 50% of the tissue culture infective dose is detected, and that value is used for the determination of the number. And here actually usually we follow the cytopathic effect in the inoculated tissue culture cells. Okay, it's widely used in clinical research and other purposes. Here also we use the serially diluted sample. So that's about this part of presentation for viral cultivation, and I hope you can follow. Thank you for listening.